Hi, I am David Degnan. I am one of three moderators or presenters this morning. We also have with us Kevin, who is our analytical chemist and actually runs the instruments. I'm the data guy, so I just process the data and ask for more of it. And Dušan also runs the instruments and analyzes the data, and he's our chemist today. So just to kind of give you an overview of what we have on the agenda this morning, we're going to give a brief overview introduction to mass spec imaging, trying to touch on the diversity not only of topics, but of the people who work in MSI, as well as all the different kinds of instrumentation. You can argue with us later if everything directly falls under MSI, but everything is directly imaging, at least on cellular levels. Afterwards, we have a group survey available now that you can take. We're kind of surveying all of the EMSL User Meeting to pull any information about what kind of tools people use for MSI, or if not, if you don't use any tools and you don't use MSI, that's fine. I'm interested in knowing what you want to learn about MSI or how you think MSI can be used in your research. So after that, we'll take a 10-minute networking break. I will go through some of the survey results after that, and then we'll get to the panel where we have these fine folk who will be here to answer questions. So without further ado, I will first talk about highlighting diversity in mass spec imaging and highlight that I believe it really is a field for everyone. Then Kevin will cover what exactly MSI is and how MSI compares to other imaging fields that are out there. Then Dušan will talk about a brief introduction to sample preparation methods. You might have noticed that there's another workshop after this from 10 to 12 where we've got a lot more details on tissue and sample preparation, and there is room in that workshop if you're interested in attending. After that, I will talk about bioinformatic tools and kind of how they fall in the MSI pipeline. It's a constantly evolving field, so you may also argue with me or ask questions about how we think about the ways we process mass spec imaging data. Then I'll also kind of cover integration of mass spec images, why we would want to integrate them with other imaging types, what kind of information you get out of mass spec imaging, what kind of information you get out of other technologies like histology, and then sort of the other interesting areas that this field is starting to bleed into. Okay, so not only is the application spaces for mass spec imaging diverse, you can see mass spec imaging in spatial multi-omics experiments where they're taking spatial lipidomics, spatial metabolomics, spatial proteomics, and spatial transcriptomics techniques and trying to integrate all of these pieces together. There's a project called the HubMap project, which is an NIH-funded project where they're trying to map every healthy tissue in the human body. And so that is one of their goals and one of the application spaces for mass spec imaging. There's also some emerging single cell applications with some other technologies that we have here at PNNL, and they'll use MALDI and other technologies to fuse the two together to get to that single cell resolution. And then there's lots of 3D imaging capabilities, the ability to not only look at where a specific biomarker is on one level, but on a 3D level in an entire system. I also like to point out that the application spaces are diverse as well as the people working in mass spec imaging. And so I've just picked a few people to highlight to kind of show you the application spaces of MSI. It's a general use technology and people are very creative. And I'm also just going to highlight a few diverse scientists. There are many, many scientists out there, some of them in our panel today. And so I want to thank everybody who is working together across the entire globe to push mass spec imaging forward. So here we have Dr. Lingjun Lee from the University of Wisconsin. She's really interested in those single cell capabilities of MALDI imaging. MALDI is a type of mass spec imaging, which we'll cover more later. She's interested in studying neuropeptides and their fundamental interactions within model systems. She's got some very nice review papers for mass spec imaging. If you just kind of want to see the state of the field and the technologies, if you go look up her at Google Scholar, she's like #1 when it comes to mass spec imaging papers and reviews. And she's also interested in small molecules and mass spec imaging for sort of systems biology applications. We also have Dr. Eberlin from Baylor University. She's done a lot of research on metabolomic biomarkers of cancer, where exactly these biomarkers live in tissues and tissue systems. She uses forensic analysis with DESI, which is another type of mass spec imaging, which we'll cover later. And she's pushing DESI into operating room kind of conditions. So real-time DESI operations to look at where specific biomarkers or where specific targets are in tissues. Very creative. She's also a developer of a bioinformatics package I will talk about a little bit later that they use to process and analyze mass spec imaging data. Dr. Fernandez from Georgia Tech, he uses MSI to do small molecule and lipid imaging. He was one of the first to push this technology in that area. He does image integration as well, which I've mentioned before, of combining different technologies for multi-omics applications. In his space, he's interested in taking MSI and NMR and merging the two together. We also have Dr. Sanchez from UCSC, and she uses MSI applied to ovarian cancer, invertebrate zoology, biofilm development, very diverse kind of interest and areas. They'll look at specific systems for invertebrates, and she will use mass spec imaging to discover things about like the notochord in worms. So this is just kind of highlight that it's kind of everywhere. There's not one box that mass spec imaging sits in. There's not just one problem mass spec imaging solves, and there's not just one kind of person who's currently using mass spec imaging. So now that I've talked about it, we'll have someone actually explain to you what it is. I will pass this on to Kevin.
So thank you all for coming here nice and early in the morning. As David mentioned, I'm going to talk to you about what MSI actually is, but it's really hard to distill 30 plus years worth of work in about 15 to 20 minutes. So it's going to be very high level and very rudimentary at some points, but we're just going to give you a nice brief introduction for those of you who don't actually know what the imaging field has actually been doing so far. So as David introduced, MSI is really a diverse field, and it really is, but there's diverse requirements for not only the ionization, but also for the detection. So you're going to hear a lot of acronyms. There's going to be an acronym zoo thrown at you today. And there's also going to be a lot of talk about what type of mass spectrometers are actually linked to these ionization sources. And this is really why I'm going to say right now that there's several large barriers of entry within key areas, but that's why facilities like EMSL actually exist in order to break down those barriers and make mass spectrometry imaging and mass spectrometry overall really accessible to a lot of broad for scientists. So in general, I'm going to show just matrix assisted laser desorption ionization, but the fundamentals are 3 key steps. You're going to have an ionizing beam, whether this is actually primary ions or a liquid microjunction, and we're going to go ahead and raster this over the actual surface. So this is step 1 for any of the mass spectrometry imaging. We're going to have an XY pixel coordinate map. And at each of these points, we're going to either fire our laser or actually drag our liquid junction over this in order to actually desorb and ionize, whether they're going to be proteins, metabolites, or even in certain cases with laser ablation, different metals as well. And at each of these points, this beam will then ionize, and we'll be detecting these by mass spectrometry. So we're taking our 2-dimensional image map and then adding 2 additional dimensions in terms of all the mass per charges of all these unique biomolecules, as well as their intensities. And then in our final step, we end up forming discriminant ion images that vary based upon their actual resolution, post-acquisition, and really show some very unique distributions. So you'll see that there's going to be a lot of mammalian tissues mixed in with this, is because clinical applications of mass spectrometry imaging really were the beginning of this. And a lot of the people within the actual field of plant science are starting to break into this and realize the potential of mass spectrometry imaging. So I wanted to go ahead and highlight just a couple of mass analyzers. Myself, as an instrumentalist, I'm more within the Fourier transform mass spectrometry in terms of the Bruker FTICRs, which are Fourier transform ion electron resonance mass spectrometers. And if you submit a proposal for mass spectrometry imaging to EMSL, you'll either end up having data that comes from one of our Bruker FTICRs, or possibly a various time of flight secondary ion mass spectrometer, or the nano SIMs as well. But there's a lot of diversity within even just the instrumentation. And each of these, depending on what you're doing, leads to different outcomes, where there's new instrumentation like multi-reflectron time of flight instruments, which are very high resolution and very fast. There's been a lot of development, like Justin Chang from Thermo Fisher mentioned the other day, of actually bringing matrix-assisted laser desorption ionization to the Orbitraps to make these more accessible. You can actually purchase these and put them on. But all of these different instruments, whether they're the Bruker Toffs or anything in between, they are quite expensive and they require a fairly large knowledge base in order to actually use. So that's why you'll have experts here at EMSL that can go ahead and actually prepare your samples, as Dušan will go ahead and mention later, but then also get you the outcomes that you're actually desiring from any of your mass spectrometry imaging. So not to overwhelm you here, but I went ahead and actually chose just a very broad amount of imaging in terms of graphical abstracts and just some pretty pictures that actually have quite some biological stories behind there. So as I mentioned before, really hard to distill over 30 years of mass spectrometry imaging research. Some of that even goes before then. But here's some really recent nice examples, and whether or not we're actually looking at intact proteins and doing native proteomics, which is something that still hasn't brought in or been able to be brought to the actual BER space yet, but we can look at these intact proteins and these actual dimers and trimers, which go up to 40, 50 kilodalton, and analyze them directly from the actual tissues and then see their very nice distributions. We can also go through and do some in situ digestions, look at these N-glycans, and this is actually a nice example from soybean root nodules, where you can actually see the actual differential abundance throughout the actual inner and outer cortex and within the infection zone within the rhizosphere. There's been a lot of development also bringing methods that you might be more familiar with, like immunohistochemistry, immunofluorescence, and using those antibody-based probes. That has actually been recently brought into mass spectrometry imaging, where those same probes now have a photocleavable tag on them. And what we can actually see is that if you look in the bottom left-hand corner, very diverse multiplex detection, because mass spectrometry is really good at detecting several hundred species at the same time, unlike a lot of the actual microscopes, which might be limited to, say, 3 channels or a couple more if you end up doing some advanced methods. There's been a lot of development within metabolic profiling as well. As one of our panelists has actually shown, a lot of work that's been put into on-tissue chemical derivatization for metabolomics, doing targeted experiments, whether we're looking at amino acid metabolism and then actually having some fundamental frameworks that are really accessible, like Metaspace, which is an online portal for data interpretation. And then we also can go through and do the same manner and go through and do these metabolic profiling and actually see all of these actual very interesting metabolites and how they interact between different spatial localizations and within the fundamental biomechanisms within there. And then we can get into the space of going larger from metabolites, not as big as proteins, but we can get into lipid imaging as well. So all the way down to roughly that 3 to 10 micron. And we're going to talk a lot about spatial resolution, sensitivity, and a lot of these very large, broad concepts. What we can actually see is that there's a grand diversity of metabolites, lipids, and proteins all the way throughout very diverse layers. And in the bottom right-hand corner, bringing it back just to a little bit of the actual NIH space, if you think of the human eye that you're using right now to go ahead and actually look at this presentation, there's diversity within the actual lipids. And in terms of the actual lipid classes themselves, phosphatidylcholines are just one of these lipids that are very important in terms of the actual end metabolism, but we can see that they're incredibly diverse in terms of not only their spatial localizations, but within these fine layers actually controlling a lot of different portions. So it's just a very big brief overview of some of the actual outcomes of the actual mass spectrometry imaging. But when we really think about how this compares to other imaging analyses and even bulk analyses as well for mass spectrometry, what we can actually do is really put them on a map. So this is actually from a very nice Nature perspective paper, but it really ends up defining a lot of these imaging modalities, whether they're going to be traditional analyses, which are more or less highlighted within yellow, imaging mass spectrometry in green, and a lot of the bulk analyses, which are really bridging the gap between targeted analyses that can go subcellular and really broad profiling, where you can have over thousands of biochemical species. So if we end up looking at this, we can see that the circles around here are colored within different regions. So we can either target RNA and chromatin in terms of either doing RNA-seq, attack-seq, or going into the actual subcellular regime with mirrorfish or all of these other seq-fish entities, which actually use a little bit more of the antibody-based imaging, which we've heard from some of our speakers before. We can also go ahead and look at some other antibody-based imaging for spatial proteomics, but these are all targeted. And when I say that there's a few well-characterized species, it really is that there are just a few well-characterized species. And there's no guarantee between sample preparations, whether or not you have a fresh frozen tissue or a formalin-fixed paraffin-embedded tissue, whether these will actually go ahead and work. So we can go ahead and use the single cell subcellular resolution, but then we can bridge this using our imaging mass spectrometry and actually detect several hundred species that actually give us a lot of broad chemical diversity, and then link this back even further to bulk analyses to really do some nice multi-omics integration. And that's really what we're kind of focusing about today within this, and just giving you a very broad perspective of what's out there in terms of a lot of the actual techniques and how we can actually utilize and what we can actually utilize mass spectrometry imaging for. So in terms of the main areas of what we can actually look at with mass spectrometry imaging, it's incredibly hard to distill all of this, but we can break it up into the fields of the omics, whether this is actually metalomics in terms of analyzing atomic emission from laser ablation ICP, which is inductively coupled plasma, very hot plasma, which breaks apart all these biomolecules. And then you can actually image whether it's iron concentration all the way throughout, or we can end up doing the advanced techniques like SIMS and nanosims, which actually end up using multi-channel detectors in order to actually detect the secondary ion beam coming off these samples at very, very high resolution. We can bridge from that atomic space back into the molecular space and do metabolomics and lipidomics, and whether this is from DESI, SIMS, and MALDI, which we'll explain in a little bit. Like I said, there's an acronym zoo out there, so I'm sorry about that. I'll keep on apologizing throughout, but we can go ahead and do a lot of non-targeted metabolomics and lipidomics, and based upon what technique we actually have, we can detect different biomolecules throughout. Bridging in even further, we can go into glycomics, which we know that glycans actually attach to a lot of peptides as well as the actual proteins themselves. And we can go through a series of actual washes and then an actual on-tissue digestion in order to actually detect these glycans. And Dušan over here is actually an expert within this. And then we bridge into one of the more interesting fields to me right now is actually spatial proteomics. And there's a lot of techniques and technologies, whether we can do the in situ digestions with trypsin, glu-C, different enzymes applied directly to the tissues that we can analyze these peptides coming directly off, or we can analyze the intact proteins themselves and actually see all the post-translational modifications, or do a targeted approach very reminiscent to immunohistochemistry, where we can actually take these probes and analyze them directly on tissue and then cleave off these peptides, which would be reminiscent and can form our ion images. So there's a very broad scope of any of this. And if you're ever interested in any of these techniques or how they can actually apply to your research, please reach out to us and we should be able to go ahead and guide you. And We'll talk a little bit more about the sample preparation for each of these in a little bit, but there is some universal sample preparations, and we can actually combine a lot of these different modalities together. So when we really think about actually combining these modalities, you might see that there's actually going to be intestinal tissue on the right-hand side. That's because within the BER space, there really hasn't been a lot of combined autofluorescence, as well as protein imaging, lipids, and metabolites, or even elements throughout there. There's only been a few labs realistically that have been doing this within even clinical research overall. But you can actually get a lot of diverse information that can actually be linked back, where proteins actually bind metals and lipids and metabolites are actually the end byproducts of the actual protein action. So a major goal has actually been the spatial multiomics, but the limitations exist because we can't necessarily do lipidomics and proteomics at the same time. And there's different sample preparation requirements for each of these as well. So doing a lot of these is actually uncommon, and there's a lot of other issues that we can talk about with actual plant research. But what the key takeaway here is that different sample preparation requirements are required for each of these tissues, whether they're roots, plants, microbes, biofilms, anything in between this. And realistically, as of right now, there's been a lot of work within bioinformatics in order to actually start integrating all of these tools together and actually giving us some really nice biological stories all the way throughout. So with that, I'm going to go ahead and actually give it over to Dušan, who's going to give us a brief introduction to some of the sample preparation methods. And when we're talking about preparation methods, it's actually how the sausage gets made, for lack of a better term. So
I can use this, I guess.
Okay.
Right?
Yeah.
All right. Okay. So yeah, good morning, everyone, and thanks, David and Kevin, to allow me to be on their workshop. Actually, so yeah, I want to talk about actually sample prep, and you will have also another workshop later today where we will go really like hands-on there, that you can see how this is going on. And Kevin was talking a lot about how sample prep is an important step, and for different application there is different sample prep actually. And to my opinion, sample prep, I mean, it's the first step in entire mass spec imaging workflow, and from that step depends all your downstream analysis. So you can have all the fancy techniques and bioinformatic tools, but if you don't prepare your sample well, actually like data that you get will not be valuable and representative actually what is really going on in the system that you investigate. So I want to say that I will mostly talk here about sample prep for multi-mass spectrometry imaging, but similar is with all other mass spec imaging technique. I see that John Cliff is here as well. So yeah, we can talk about nanosims and those types of samples as well. But what I want to say is that very few samples you can analyze just as is. Maybe we can analyze like leaf surface or flat samples, even like when we go in the analysis of microbial interactions, we put that samples on the agar plate and analyze them directly there. But usually, especially if we go in the BR space and analyzing the plant systems or different plant organs, actually it's very important that we need to embed those samples. So this embedding is, and you can see here like three main reasons why embedding is really important. First, basically, if you want to obtain like quality measurements, your sections need to be at the first place very flat, and at the second place they need to be like relatively thin due to, and we can talk later about that in the deeper why they need to be thin. Lot of instruments require that you have like conductivity there and transfer ions to the mass spec. So we are limited on those samples that are roughly sections 5 to 30 micron ideal. And as it mentioned here, it really influences sensitivity. So you can imagine that maybe from the bigger sample, you can actually get the more material, but actually all those techniques are mostly like surface techniques. So we don't actually go down there in the bottom of the sample. Then also, especially in the, you can see here, I will go on the I mean, we already, Kevin, I know that pointed in the second one, let garbage in, garbage out is what we call this second reason why we want to embed and section well, and it is that we want to basically preserve morphology of the sample, right? And then in the plant tissues especially, there is a big problem comparing to the, let's say, when we section like mammalian tissues, they're quite easy to section even without embedding, right? They have a lot of fat there, and you just like smooth going to their sectioning. I mean, I'm talking about like, let's say, kidney, liver, not about bones and those kind of things. But in plants, a very big problem are those huge vacuoles. A lot of water content, lot of empty space, and then all of that will cause us problems in sectioning. We'll have like entire sectioning workshop where you can really see how embedding going on and basically how we do it experimentally. But main thing what I want to highlight here is that we want to preserve morphology of the sample, but also what is really important is that your sample prep depends on your application and what kind of molecules you want to see. So it will not be the same sample prep for this sectioning part. It will be the same if you want to analyze metabolites, proteins, and so on. But downstream, we will not go in downstream preparation. Kevin hints something there, but we can also talk about that. So you need also to think about future application. Can you go on the next slide? Yeah, okay. So I also want to tell you that we usually analyze here you as an EMSL user sample, EMSL users. If you ever want to send us samples, we need to talk deep. Like if you already have experience in sectioning, we also need to provide you conductive slides, usually like with a requirement for example, multi-imaging. So you can just, if you're experienced actually and know how to do it, we can provide you slides. You send us those slides back. They need to be as well vacuum sealed and put back to us, and we can talk. I said if you're really interested in that, we should go and on that next workshop where I will cover that more deeply. If you want to see how what kind of samples we accept and how that this is more about theoretical details, what is necessary for good and why it's important to do sample prep on that way. So in ideal world, embedding medium are usually like some kind of polymeric substances, either kind of polypeptides or like gelatin, for example, or polysaccharides. A lot we use as a embedding material, and in ideal world, this embedding material should enable multiple modes of either mass spectrometry imaging, and of course we want to be enabled to do microscopy. After that, we can overlay ion images with histology, right? And then, as I already mentioned, the most important thing is to preserve tissue integrity. You know that in some cases, probably people who are from clinical world, we use like formalin fixed paraffin embedded samples. They are probably the best for preserving tissues, but they are not actually ideal for small molecule analysis, and they are completely not ideal for lipid analysis. So because actually, like it is just way how they're prepared in the different organic solvents, where a lot of those, let's say like hydrophobic compounds are already washed out and we cannot analyze them. So, but for example, for Kevin work or in the protein field, or like for post-translation modification, like glycosylation, those samples are actually like very useful, and we use them as well. And then the most second thing is that we don't want that this embedding material cause delocalization, right? What Kevin pointed out right here, because like the most important thing in imaging is actually that you want to have like information about localization of your molecules. So if you have the localization that is bigger than your step size that you use, then you don't have valuable information. And also there are a lot of matrices or embedding material that are perfect for sectioning, but they provide like very significant signal suppression. Probably the most important and probably the most common of those is like OCT, like optimal cutting temperature medium that is used for like microscopy preparation, but it's actually completely not compatible with mass spec due to, sorry. This is my time to wake up actually. Yeah, and a lot of those some of embedding materials have like a lot of additives that are there, or polymers that kind of just like are so abundant in your mass spectrum that basically are not useful there. And as I said, like OCT is probably the most known for that. And as I said, like number 5, we don't want to use any kind of especially in the molecular imaging space, for example, for the SIMS and other technologies that are not providing exactly like molecular information, but more on the elemental point of view. You can use also like chemical fixation, but we don't want to have any uncontrolled changes of biology, of biomolecular chemistry. So we don't want to do any crazy stuff to make mechanically stronger our tissues, because like we will use information about molecular types. And it is some brief overviews. You can go on the next slide, Kevin, please. So this is something that we did here and investigate in the plant space, because there are a lot of work really, as Kevin mentioned, in mammalian space, especially because, as Kevin mentioned, like mass spectrometry imaging found like really great application there from clinical point of view. So we want to accept those ideas and implement it in the plant world, in microbial world as well. And so we usually, like, of course, plants are composed from the cell walls that are polysaccharide in essence, celluloses. So most of embedding materials that are using for preparation of plant tissues are kind of modification of the cellulose, that polymer that we use. And then there are different ways how you can we prepare, for example, several different embedding materials, and then try to section this is popular route. And like we don't want to have any damages by sectioning those holes and gaps that will basically destroy cellular content. You want to have like really nice and preserve samples here. So like in this one, like M7 that Kevin is showing. Okay, we can go on the next slide. And also like this is what I was talking about, the localization of the signal. You can see how we don't want to have any kind of smeared images. It actually says that this in embedding materials, molecules, especially when we deal with the small molecules, they leak out. So it is something that we want to avoid, right? We want to have like sharp edges to have like really that our ion image like overlay nicely with the histological information in the tissue. So that is something that is goal of any embedding material preparation. And then on the right-hand side, actually, you can see that we also want to increase sensitivity, that we don't want to have any suppression of signal. So it is also what you need to be careful of. And with that, I think, yeah, sure, please, if you're interested, go on the next. It is at 10:15, and there will be a few of us that will guide you through the sample prep, not only for the mass spec imaging, but for general like microscopy and other like laser capture microdissection will be shown there. So if you're interested for those kind of like nanopod sample prep, yeah, please. There are still, I think, spots remaining in this workshop, maybe like 4 or something like that. So yeah, please sign up. Yeah, I don't know. We want the questions now or after or okay, yeah, well, after.
Yeah, we can take them after within this, and we're going to throw a lot of information at you just really quickly within this, but it's really giving you just a broad perspective of what the differences are and how each of these techniques has a lot of work that goes into this, where you can take your plant root, but you have to go ahead and actually prepare your plant root in several different ways for several different methods, and then you have to get them back and actually organize a lot of them into these bioinformatics tools. So right now, I'm just going to go over a brief different types of mass spectrometry imaging. So I mentioned a couple within the actual showing the pictures of the instrumentation, and it's always nice to see what the instruments look like. They might be scary or complex for some, but in terms of what we actually have here and why I say that I was going to throw an acronym zoo at you is because if we think about molecular imaging or even just atomic imaging, we have a various range of where we can analyze in terms of spatial resolution down to the nanometer scale, especially for the secondary ion mass spectrometry and nanosims, which is a big capability here. But also every single one of these we actually have within EMSL. So not only can we do the laser desorption ionization, which allows us to go ahead and actually profile a lot of the actual diverse chemical compounds from soils directly intact, very akin to doing like the atmospheric pressure photoionization or APCI, which is the chemical ionization. But we can also do the matrix-assisted laser desorption ionization. And we've talked a lot about MALDI. You've heard a lot of talk about MALDI as well. But the very interesting thing is that it's not the most sensitive technique, but it's so versatile in that we can actually analyze nearly every single biomolecular species directly by MALDI. You can actually analyze things up to a megadalton directly by MALDI, but you're limited by your actual detector. And if we end up thinking about some of the actual ambient ionization, which is actually going to be happening outside of vacuum. So the one thing that I haven't mentioned is that SIMS and nanosims actually requires ultra-high vacuum. Laser desorption ionization can either be at atmospheric pressure or it will actually be at an intermediate vacuum, roughly that 3 torr or a couple of millibar within this. But then we can also do things at atmospheric pressure, atmospheric temperature. And this is where we have the nanodesorption electrospray, desorption electrospray, the liquid extraction surface analysis of, has anybody here used a pipette before? So you can imagine actually pipetting and aspirating your liquid over the actual sample, but then the LISA device actually takes that and directly injects this into the mass spectrometer after the fact. So it's more or less just a manual process, which a lot of these things, what they came from fundamentals of just liquid solid extractions, liquid extractions, whether they're sputtering or thermal desorption. We can see that there's several dozen fundamental mechanisms which we actually have, and I think in terms of the actual field right now, there's about 120 different ambient ionization techniques. And this is where you can get as crazy as you want, but there's a lot of them that you can actually use for different purposes. And you can actually analyze whether they're viral capsids or actually going into the direct organelles of a lot of these microbes, or even within some of these ambient ionization, just profiling single eukaryotic, prokaryotic cells as well. So ionization techniques really govern the spatial resolution, but also the detected biomolecules. Doing anything like intact protein analysis from SIMS, you'll have to go ahead and put a tag on there and actually do things like either with a MIB-TOF or antibody-based reagents. Doing actual atomic imaging from nanodesi, not really feasible at this point in time. But there's a lot of things that you can actually do by combining different methodologies and really spanning that scope of omics. So this is going to be a very brief slide. I'm not going to go ahead and talk about the fundamental mechanisms of each of these. But as you can actually see in that first slide, I did not lie to you. There's 3 major steps. There's always going to be whether or not an ion beam, an ultraviolet laser, or a mid-infrared laser, that we're going to go ahead and actually ablate that tissue. And either that laser itself or that ion beam will then ionize these molecules which are coming off, or these atoms, or we'll have a secondary ionization that can end up coming in after the fact. So like I said, there's a lot of acronyms within this, but when we talk about the actual plant tissues, and as Dušan had mentioned, there's higher endogenous water content. You can think that putting something at ultra high vacuum with a lot of water inside of it, it's not going to be a good time for anybody. But also the one thing that we have to think about is that plants typically have a larger cellular size, where a lot of these fungus and bacteria, they'll actually be submicron within this. But when we talk about plant cells within, say, a soybean root nodule, we might be upwards of 75 microns for one of these actual infected cells with bacteria inside of it. So what that actually means is that there's normally a lower molecular content per each cell, and we also have a thick cell wall that actually ranges based upon whether or not there's abiotic or biotic stress. So a lot of these different methods have different utility, and there's a leaf shown for each of these, but in order to actually get a leaf into ultra high vacuum versus mid vacuum or analyzing it just ambiently with some of these different techniques, it's very, very hard and very different. So that's where, you know, the sample preparation, 90% of the time really should be put into the sample preparation, and then 10% of the time within the actual imaging and the outcomes from that. So in talking about some of this imaging, this is actually some beautiful images of an actual root cell, a root that's actually sectioned that has a biofilm that's directly around this. And what this is actually analyzing is actually being analyzed by nanosims. And this is actually courtesy of John Cliff up here. And he'll be able to talk a little bit more about this because I'm not an expert with the nanosims. However, what we can actually see is that we have very fine subcellular resolution, and we can do very nice quantification of different isotopic tracing experiments too. So if you're ever interested in doing different single cell analyses or actually doing pulse chasing experiments, they can actually do this within the nanosims. So not only can we actually see subcellular, we can actually see the uptake of different nitrogen isotopes or different carbon isotopes directly by this actual technique. So this is actually a great case study for if you're interested in these isotopic tracing studies and you need that fine resolution. The SIMS and the nanosims is actually really ideal for this. And we're just going to go through a little bit of each of these to show a nice little case study for each of them, but we can always talk more about this later, especially during the panel. MALDI, like I mentioned before, is one of the ones that I primarily work with as well, but we can have a case study for this on-tissue chemical derivatization for this increased metabolomics, but we can not only can do metabolomics, lipidomics, and spatial proteomics, we can actually start doing profiling the cycles, the amino acid metabolism, looking at the TCA cycle, seeing how energy is actually transferred. And if we're thinking about different plants and microbes, carbon sequestration and nitrogen uptake in terms of a lot of this, getting at these real biomolecular mechanisms under nitrogen fixation or anything in between. And we can end up having pretty high confidence within this actual metabolic profiling, especially adding these tags to them. Nanodesi is actually another one where if you think of two straws, you're actually pushing a liquid through, you're forming that liquid junction, and then you're taking that liquid junction and then ionizing it into the mass spectrometer. There's actually a case study done at PNL a while ago, but you can see that this is actually a two-dimensional or three-dimensional biofilm that was actually grown on an agar plate. And in terms of most of our mass spectrometry imaging, it's done within 2 dimensions. But within constant distance mode, where we're continuously moving that microjunction based upon the topography of this, we can actually relate this back to the actual topography of the actual biofilm, seeing how all of these surfactants and polypistatins actually change in terms of their intensity and distributions throughout the different areas of the actual biofilms. So we can take that mass spectrometry imaging from a two-dimensional space and not only break this up and do multiple reconstructed three-dimensional images, but also look at the actual topography with things like nanodesi, or especially secondary ion mass spectrometry. Looking at DESI, there's a case study for actually analyzing these high water content samples, including different leaves, where this is actually a nice case study of profiling the phytohormones, which are incredibly important for biotic and abiotic stress responses. There's some acronyms up here, but this is ascorbic acid, salicylic acid, jasmonic acid. And what we can actually see is within this wounded region of the actual plant, we can see that there's actually a stress response that goes through these hormones. And not only can we go ahead and analyze them based upon the intact mass, high resolution intact mass, based upon their mass per charge value, but a lot of these constant techniques like DESI and nanodesi actually allow us to go through and do this tandem mass spectrometry, where we're taking these molecules and we're adding energy and breaking them up, and then we can identify them based upon their characteristic fragments. So really high confidence imaging outcomes can actually happen from this. And finally, another technique that we can actually do here is actually the laser ablation electrospray ionization, which acronym again, but we can go ahead and do some very interesting targeted single cell metabolomics. And when we think about this, we're showing a picture here. This is actually from a soybean root nodule. There's one cell here, and then after the ablation, you can see that that tissue is actually gone. That tissue is then subsequently desorbed and ionized, and we can detect from these single cells all of these different biomolecular compounds. And this is where we get into that single cell analyses, where not only can mass spectrometry imaging give you these fantastic ion images, which are very characteristic of the actual morphology and the anatomy, but we can go through and actually see how each of these cells, whether they're infected with bacteroids or actually just control cells, how the metabolic profiles actually change. And this can give us a glimpse into how nitrogen fixation is happening, as well as just the overall amount of variants that are persistent within these single cells. And then finally, another thing that we have actually done here within EMSL is actually use this liquid extraction surface analysis. And as you can actually see, the resolution is definitely, if you imagine taking a flip phone picture versus an actual very nice high resolution camera, which we compare Lisa DeMaldi. But because we're doing that pipetting onto that surface, we have several minutes of ion current, which we can work with. And we can do very high confidence tandem mass spectrometry, or MS2, where we can actually match back up all of these different distributions, whether they're within cyanobacteria, fungus, moss, or these tripartites. And we can actually see that there's a broad chemical space where adenine can be actually detected within a couple of these and some not detected in others, but we can actually start doing these molecular profilings within this. So that's really the brief overview of the types of MSI. We could probably spend a whole entire day doing this, but I'm going to hand this back over to David, and he'll tell us how we actually go ahead and take some of these images and go ahead and actually work with them in terms of bioinformatics and image integration.
Okay, so I know we're running low on time, so I'm just going to briefly go through this. So we talked a lot about different mass spec imaging techniques and sample preparation. And here I've constructed a pipeline, not the pipeline for mass spec imaging. And I've just put some tools that get used at each of these steps, whether we're doing pre-processing, I'm thinking about noise thresholding steps, statistical filtering, peak picking, applications to feature identification where we're looking for specific regions within a tissue of interest, to the data visualization step. And I think when people build things like this, you might immediately think like, oh, one tool is better than another. I want to point out that like Vitesse is an excellent visualization tool. So please don't think I'm comparing these tools. I'm just saying the steps of the pipeline that we tend to use these tools for. And so these tools can either be in green. This is things that we code manually, or there's a package for, and blue, or this purple color. I'm not great with colors. These are all like GUI or graphical user interface applications that people use to process this data. People who tend to build custom scripts are trying to get away from what a lot of these tools do, which is each of these steps manually. For example, for feature identification and extraction, With Skills Lab, I believe, it's a manual dragging and dropping of each of those features that you want to pull out and draw on. Whereas with Cardinal has some applications. It's an R package, and Cardinal MSI is a tool that's being developed. They have some unsupervised segmentation. So what they'll do is they'll take those regions of interest and they'll highlight it for you. And so that's kind of the direction that these tools are moving, is more automation, especially as Dušan and Kevin keep giving me more samples that I have to analyze. So that is kind of the state of bioinformatic tools. MATLAB is super great if you're new to coding and want to start somewhere. For doing image processing, it is not open source. So if you want to do something that is open source, OpenCV is a Python, one of many Python imaging libraries, but it's worked pretty well for some application spaces in MSI, which is why I point this out here. And then when I think about where we are and where we're going, a big step is mass spec imaging integration. We've talked a lot about how when we get these mass spec images, we get the abundances of biomolecules essentially across a tissue, but then what we lose is feature information from histology and other pieces. So that's why people like to, what they do is they do this step called co-registration, where you have your histology image or your image of interest, and you have your mass spec image, and you align the two images, the imprint of your tissue. And then once you have those aligned, you can do the next step, which is called segmentation, where you pull out those features and you put them together. So that's what we mean when we're talking about image integration. That is just one approach. People are also putting immunofluorescent images and mass spec imaging together. Any sort of imaging technique where you get a tissue, you can technically integrate. So this is a very obvious area of development: autofluorescence, confocal microscopy. We're going crazy out here. So there's lots of different techniques. Another one is to use these softwares to identify these regions, like I was mentioning before, in the construction of 3D images. When it comes to segmentation, here's just 4 approaches that I found, but if you Google segmentation approaches that are being developed, not just for mass spec imaging, but through Google and Microsoft, there are over 40 algorithms that I was able to pick up and list by hand before I got tired. So there's a lot of options out there for segmentation and for detecting features of each of these tissues. Then more statistical bulk omics approaches. Sometimes we just want to compare if this is a specific biomolecule in the picture here, and we've got 2 regions of interest. We want to just compare if there's specific biomarkers of interest that are differentially abundant in 2 different areas of a tissue. And so this is also a potential end goal of segmentation. Once you have these regions and you have the mass spec imaging, then you can actually move this along and do statistical analyses where you actually compare the differences between regions. So that was very fast. You can ask me lots of questions later. I'm just trying to keep us moving. So I think very big picture, if you're like me and you like those big pictures, mass spec imaging has diverse application spaces and goals. And mass spec imaging in plant science, as well as other different areas like cancer biomarker discovery, is emerging and continuing to develop. There are many different types of mass spec imaging with different spatial resolutions and biomolecular coverage, and it's constantly growing and evolving, and it has a lot of untapped potential. So when you're thinking of a specific problem and you want to try a new approach to your problem, maybe we can help you be creative. So we're going to, oh, yes, we can clap. So during this time, we're going to have questions until 9 o'clock, which gives us about 8 minutes for any questions. Feel free to do the survey, and then we'll take a 10-minute networking break. I'll go over the survey results, and then we'll have our imaging panel.
And this is actually going to be the link for that survey too. So it'll be tinyurl.com, EMSL MSI. And the results will be displayed in real time. David will go through that. So if you have the time, please go through and do that. But if there's any questions, we can go ahead and foster and answer those right now. So there's a lot of information we threw at you, very glossing over a lot of different portions of the field, but please let us know if there is anything.
Hi, thank you. I'm kind of curious, what does the data size and computing power look like for this kind of work?
Yeah, so I can comment on the data size right now. Depends. It can be in terms of megabytes, but we actually have an imaging run right now of, if you've heard about the terraforms, we're actually imaging that entire membrane off of the actual rhizochip from a poplar root that was actually grown within this with endophyte. And that's going to be about 1.1 terabytes of raw data. And when that actually gets processed, you're now processing that raw data into either skills lab or different pipelines, creating downsizing, hopefully, but maybe another 500 gigabyte data set. And eventually the images are very tiny. We can compile them and compress them into different forms, but it can either be a couple hundred megabytes or it could be a couple terabytes actually in terms of this. And David, I don't know if there's anything else to add, or Dušan, in terms of that, but yeah, it can be big. So that's where a lot of the actual imaging data, if we're doing like an interlab study or sharing data with you, getting you the actual raw data, there's not a lot of good ways in order to ship a 1 terabyte file across the country in terms of that. Even FTP servers can be somewhat limiting. But the raw data, it also can be behind some paywalls, because a lot of those premium software packages can be several $1,000s a year. But that's something that we can actually get these into open formats and actually share them online within portals like Metaspace, which you don't even have to worry about that. It's an online visualization tool. And there's been a lot of work within David, and Lisa, that they showed the Trelliscope displays, which are actually just HTML links. And we can do all the visualization directly within that. So could be several, some of these computers also could require, say, 100 gigabytes of RAM in order to run properly, but even a laptop can run a lot of these softwares. So.
And the data that Kevin is talking about includes the scalars, and that's why it's so large, is there's all those different scales to it. The file format that's emerging, that's becoming more popular, is these OMI-TIFF files, because it can include both the mass spec images and the biomolecule annotations all in one space. Those tend to be smaller, like around 20 megabytes, which is something I would rather work with. So that's kind of like the difference, I think, in the data size there.
And there are also like those universal, ICML files that we can always they can be shared between they're much smaller in size. So yeah, because you can think about that as well, like in terms of sharing data. And also what I want to say, we use here FTICR's instrument that, are so much details there, and to do in transients capturing all kind of things is so huge. Right. So I guess if you use TOFs or other stuff, data size is much smaller.
Yeah. That's something that in terms of the SIMS aspect, I don't really use a lot of those, but I mean, think of the FTMS. For me, the actual shape of the peak and how it actually is represented in the spectra gives a lot of information, but some people only care about the actual centroid, which gives you the actual M/Z and the intensity of this. So it kind of all depends, but we can always bridge a lot of different formats within this. So.
Okay, thank you.
Yep, thank you.
I think we have time for one more question, if there's any.
So I'm kind of new to this whole thing. And I really didn't, kind of came in a bit late, but what do you actually use to mount the sample? Is it plastic or
So it depends. I can give a little brief introduction. We can either use things like carbon tape in order to actually get them onto the slide. There can actually be conductive copper tape as well, or a lot of these sections are actually just thaw mounted. So they're actually being sectioned at -20, and then they're actually mounted directly on the slides from the temperature differential. And
that's about it. Yeah. You can be creative as you want, right? So we usually tau mounting is usual way. Some people use also some other tapes, like cryogen tapes and other things. Yeah, different labs use different things, but we use a lot for samples that will not adhere well to the slide. We use copper tape, as Kevin mentioned. In our case, especially in MALDI imaging and mount it off. And I don't know why in FTICR we need those conductance though, but okay, that's matter like to don't go in detail there. Yeah, you need to have like conductive surface as well. So that's also kind of thing that you need to think about. So why we put those conductive tapes there as well to mount the sample, but yeah.
All righty, thanks.
Thank you.
Okay, we'll take about a 10-minute break and see you all back here at 9:10. At the panel today. So it looks like a majority of the people have none or some experience with mass spec imaging technology, and we do have some experts here, maybe our panelists. We had a question here too about if you do MS research, which kind of omics do you tend to do? It looks like majority of people don't do mass spec imaging research here, but we've got some lipidomics, metabolomics, multiomics, transcriptomics. I'm sorry, Dušan, no glycomics, but it's okay. We have some answers here about what people's goals would be with mass spec imaging, which I will use for my own purposes, but I appreciate you guys filling out these answers when we design tools that are generally applicable. It's nice to know what people want to do with mass spec imaging when we make these tools. Looks like most people here use or are familiar with NanoDesi, MALDI, and SIMS, which is great because that's what our experts are familiar with today. Most people seem to fall in the data acquisition part of the mass spec imaging pipeline, though we do have quite a bit of representation from people interested in visualization and interpretation, which is more my area. So that's exciting. Comes to file formats, it looks like those IMZML, IBDs, and OmniTIFs. Actually, I'm surprised anyone's using them. This is very much a new approach for mass spec imaging. So that shows its potential as a file format, ASCII as well. And then softwares, it looks like people are using Cardinal, MSI Reader. Hard to tell with an N of 1. Please don't tell any statisticians. But basically, it looks like these are the kinds of areas that somebody's at least used. When it comes to barriers, it looks like things like the size of the data, the sampling techniques, having too many unknowns in microbial communities, general inexperience. What a fair and honest answer, whoever put that. I appreciate that answer, and data processing. So I'm going to pass this over to Kevin to moderate the panel and to get our panel members down here.
Fantastic. Great. So I think, let's see. I think we can try to get this presentation back up and going. Might need a little help, but we have the pleasure of having 3 panelists today. 2 of them are internal to P&L with an EMSL, and then one also external who's a very experienced mass spectrometrist as well. So I'll wait for them to come up here and then we'll go through and actually introduce them. So yeah, whichever place you want to go ahead and actually sit down. I think we have 1 or 2 microphones. I don't know if this is actually working as well, but we can pass that along. I don't know if we have a microphone for young Jin. Or you got one for questions? Okay, here, I'll pass that to young Jin. So each of you guys can get your own microphone. Perfect. Okay, so lots of experience sitting up here, so hopefully a lot of these questions we can go ahead and get answered. I'm going to go ahead and actually have you guys do a little self-introduction if that's okay. And we'll start with Kristen, and then we can go down the line all the way to young Jin.
Yeah, so
well, yep, so I can read these to you if you want to.
So just introduce ourselves, and then second is describe our technologies we use. So I'm Kristen Burnham Johnson. I've been at EMSL for about 15 years now, and I'm the metabolomics team lead. And I came as a postdoc originally. I was just going to stay for a few years. And really, when you think of the pictures of all the instrumentation and all the experts that come along with EMSL, it's a very difficult place to leave. So 15 years, I'm still here. I'm still very happy.
Fantastic. And do you think you can briefly mention like what you've actually used mass spectrometry imaging for? I know you've done some very fun things with fungal
So I
gardens.
actually, back in 2003, when I was choosing what lab to do my PhD research with, I learned about imaging mass spectrometry. So I specifically chose to go to the professor's lab that developed MALDI imaging mass spectrometry. And I think the power of it is, you can see, if you think of those pictures that they showed with the images, those are micron-sized tissues, and you saw all the diversity that's in there. So really, by being able to see everything spatially localized to different things, that's the power that EMSL is offering. So if you imagine you just crust up that whole tissue, you homogenized it all up, you're going to dilute out your small, really important factors that might only be in little tiny regions, and you might not get the answers that you want because you're going to mesh everything together. So spatially visualizing things. Of course, I'm a huge fan of MALDI imaging. It's one of my favorites. And then also with DESI, and you can use all these things that these guys have developed to see specific types of molecules. You can enrich for them. You can see the differences and get that spatial localization and the co-localization. So that's what I think the power is you can see lipids, metabolites, and proteins all within a little tiny region. And that's where you can get the biochemistry insight. What's my pathway? What's turning on?
Fantastic. Thank you, Kristen. I'll go over to John next. If you can please introduce yourself and what technologies you use and some systems. It'd be fantastic. Sorry about that.
I'll probably mess this one up too. No, I won't touch it. My name is John Cliff. I've been told that I was the first external user ever at EMSL. And at the time I was studying nitrogen cycling in soils. And I was the first person to put an in situ silicon chip in soil. And I analyzed it with TOF SIMS here at EMSL. And because I'd actually touched a SIMS instrument, I was hired on at National Security here in 2004 to measure, to try and estimate how much plutonium certain countries were making. And I can't tell you what countries those are. So I've been around the world. My specialty, my passion is still biogeochemistry, but I do pretty much everything, and I'm a nanosims expert, although up here I just put sort of like, I didn't put expert for myself. That's a hard word for me to use. So yeah, so I really like biogeochemistry. It's sample preparation, as Dušan was mentioning, is really the hardest part in all this, and it's something that we're continuing to research. Anything else? I think that's,
I think that's good to start off with. We'll pass it off to Youngjin.
I'm Youngjin Lee from Iowa State University. I have been working on mass spec for like 30 years, starting from fundamentals, developing ion mobility mass spec and proteomics. And when I joined Iowa State 15 years ago, I have been working on mass spec imaging, especially with MALDI. I have been mostly working on plant metabolite imaging because we don't have a medical school in our school, but there are a bunch of plant people around. And I happen to like plant, I guess.
Oh, that's a good answer within that too. A lot of people here are actually fundamental to actually bringing a lot of this to the BER space and actually doing a lot of that actual plant science, which it's not as common. We're a very small niche of people, but that's where if you do have any of the interesting problems or questions within this, we can really hopefully address a lot of that. So in terms of our next question for the panel is a directed question. After this, we'll go through a couple of these, then we'll open it up to the audience. If you can go ahead and ask whatever you want of the panelists within reason. So right now, question number 2 to each of you, and we'll go in the same order from Kristen to John to Youngjin. What do you see as a major advantage of using mass spectrometry imaging over other imaging technologies, whether this be just normal microscopy or immunohistochemistry or anything in between?
Yeah, and also I didn't follow instructions completely, I think, but discussing what types of systems I look at. So my favorite thing to look at right now is really complex microbial communities that are made up of fungi, different bacteria, really working together in symbiosis to perform tasks that Department of Energy cares about. Lignocellulose degradation, who's secreting the enzymes, what's the bacteria doing, and how they're all working together. And so what was the question? How are we use? Oh, how is it over other? Yeah, this one's a easy one because it's untargeted. You don't have to know what you're looking for. So you can just look at your space and see what's interesting. So if you're looking for, lipids, if you could use maybe immunohistochemistry to look at different head groups. If you do lipidomics imaging with MALDI imaging, you can actually see what class of lipids are, as you saw in Fantastic Talks, and then also the fatty acyl chains. So with plants, what's a really cool thing if we think of jasmodic stress signaling? We saw the cool lipid images from our talks where it said, okay, a lot of these things are enriched in 18:3. That's alpha linolenic acid. So why do we care about that? Because a lot of times when we first started publishing these papers on lipidomics imaging, people go, who cares? Structural lipids, they're just in the membrane. They do nothing. We don't care. And it was really hard to get some of those out. Well, now we've gone past that and we said, they're not just structural. These fatty acid chains, they're the primary things. They're actually cleaved off. They're metabolized into those effector molecules. So 18:3, it makes jasmodic acid. So it does it through this full pathway. And it can also make important signaling molecules non-enzymatically. So we're going beyond just looking at proteins where we're saying reactive oxygen species, they're knocking off these fatty acids, they're turning it on as quickly as possible to make the effective molecules that are going to respond to stresses, pathogens, droughts, all of this. So we can look at a system that we're interested in, and we don't have to go in with the knowledge of, I'm going to look at this one protein and I'm going to see what it's doing. We can combine it with immunohistochemistry to look at cell types, to get other information and overlay it, but we don't have to know that information going in. You have a cool system. You want to see how the lipids, metabolites, and proteins are changing. You have that ability with the EMSL tools that we have.
It's a very good answer within that too. And I think those fungal gardens that Lisa showed yesterday too, it's very hard to actually do immunohistochemistry on those because there's such a broad amount of tissue types, and whether they're microbes and fungi, as well as the actual ant leaves that actually could be within this. So very hard to actually do targeted techniques when there's 17 different types of things within these complex samples. So John, I'll ask you the same question. What do you think an advantage of mass spectrometry imaging is that you've used over anything else? So especially within terms of like the TOF sims and nano sims.
I guess the main advantage is the chemical information you get at such a small scale, potentially. TOF sims tends to be non-targeted. Nanosims, we have to know our target. We have seven detectors that we can use, so you have to know what isotope. Where I see the advantage here is, so we're really having a hard time doing in situ work. Like how do you keep your sample spatially intact, keep that integrity? And we haven't figured that out, but that doesn't mean you can't set up directed experiments to test hypotheses at these scales. And when I was a student here, I did some experiments where I put say example, manure on one side of a chip and straw on the other. And you saw this amazing change in the amount of ammonium that was assimilated just like within a micron, and so you can do experiments like that. One thing that I've seen recently, and this is probably hopefully going to go out for review here in a month or so, is somebody put carbon-13 labeled glucose into a soil, came back a year, and there's still carbon-13 there. So that kind of surprised me. But what was really cool is, and this is something that NanoSim showed us, is that the carbon-13 that was labeled was correlated to nitrogen. So it wasn't fermented or turned into a polymer. Well, this is a hypothesis of mine, but it was put into recalcitrant compounds like chitin, cell walls, or things that are absorbed strongly. So I don't know. I don't know if that answered the question, but something I wanted to talk about.
No, that definitely does, actually. It's a very unique perspective too, because bridging from a lot of these larger molecules just to the atomistic portions, you can get a lot of information just by doing these targeted experiments that you can't necessarily do unless you're actually looking at these isotopes and doing these isotope tracing experiments. So that's very, very nice. Youngjin, I'll ask you the same question. What brought you to mass spectrometry imaging 10, 15 years ago? And what do you think is the biggest advantage?
On top of the chemical imaging and untargeted analysis that already talked about, I think another big advantage is multiplexity. So if you're doing optical imaging, let's say fluorescence imaging, maybe the best colors you can label might be 5 or 10, but you cannot image like more than 10 compounds at the same time because of the bandwidth of the light. But mass spec imaging, the mass dimension is huge, and especially if you're using high resolution mass spec, you can simultaneously image in principle up to hundreds of thousands of compounds at the same time. So that's probably the most multiplex imaging only mass spec can do.
Yeah, and that's actually where like a lot of these methods bridging into the antibody base, bringing it to the actual mass spec detector instead of actually using the microscope, because it is such a powerful molecular microscope in its own sense. So it's actually a very good perspective within that too. So with that being said, I'll bridge into our next targeted question. And actually, you know what? I'm going to go back and I'm going to pull an audible. Question 2.5. What do you think is one of the biggest disadvantages of using mass spectrometry imaging as well? Because we shouldn't always just be talking about the pros of this. There are also cons to a lot of this as well. I don't know if you can give a couple of words on that.
I think one thing is your depth of coverage. A lot of times, if you're looking at something like MALDI or a surface sampling technique, you often don't have that extra separation. So something that's really low abundant might not be detected with enough confidence for you to report. But the technologies have evolved. So, we mentioned ion mobility mass spectrometry. So, if you think of a traditional, maybe proteomic experiment, you take your sample, you homogenize it, you digest it into peptides, then you use liquid chromatography to separate those peptides, because the idea is you want everything as isolated as possible so that you can see that peptide, you can fragment it, you can identify it. So if you're doing a surface analysis, you don't have that extra separation. So we're starting to advance the technologies of, okay, well, maybe we're not going to do liquid chromatography, but we're going to do a gas phase separation. Or Kevin talked about DESI, where you're using that straw and you're feeding it solvent and you're sucking it up. Well, we have ongoing expertise here where why don't we incorporate a little column in there? Why don't we include a little separation? So as our sensitivity grows, as the expertise grows in the sample prep, we can get around some of that. We might not get that same depth of coverage as we do if we just homogenize everything, huge bulk samples, do all the identifications. But you get that really nice spatial resolve pattern, which you're not going to get with other methods. So we're overcoming the limitations slowly together.
That's actually a good point too. For a lot of you that haven't done things, you can expect from the nanopots, LCMS, which is one of the most advanced technologies that we have here for the proteomics, you might be able to detect 6,000 proteins based upon the actual peptides that you can actually see. If you go through and start doing top-down proteomics and TAC proteins, you might only see 500. And if you start doing the actual imaging of this, you might only see a dozen or a couple of dozen within this. So when you think about doing a lot of these techniques together, the scope and scale of them really starts to go ahead, and you might be targeting one specific pathway, but we might not be able to get you that specific pathway unless we do some really advanced experiments. So same question to you, John. What do you think the biggest disadvantages of, say, nanosims and anything in between?
Well, all of them, it's dollar signs.
Yeah.
The cost, there's a $17 million magnet down there, and that has nothing, that doesn't even have the sample introduction units or the amount of data that comes out. And it's like that's got a really big price tag attached to it. A new nanosim today, I asked yesterday what it would be. Still don't know, but $8 or $10 million. In my case, sample preparation is a big deal, and they have to be under we have to work at about 10 to the -9 torr because we've got, 8,000 volts in a space of less than a millimeter. And you have a tiny little bit of gas in there and you could start arcing, and then you've got a week of fixing and tuning.
Yeah.
Just because of that. So there's, all these huge advantages come at disadvantages. NanoSims in particular works right at the edge of physical everything.
Yeah.
Voltage is, we're counting single atoms, and it's, one little thing breaks and the whole thing goes south, and there's a lot of maintenance involved in just getting day-to-day data.
Yeah, absolutely. And the breakdown voltages, you're limited by vacuum, you're limited by a lot of the fundamentals of the technique, so that makes a lot of sense. Youngjin, I'll ask you the same thing, and I think you have a unique perspective as well, because you're trying to actually do a lot of these isotope tracing studies with MALDI and FTMS detection overall. So I think you have a pretty unique perspective that you can go ahead and give in terms of some of the disadvantages for this.
So some of the disadvantages that mass spec imaging had, some of them may be potentially overcome over the years, like sensitivity and so on. But there are some fundamentals that there is no way mass spec imaging can overcome. One of them would be, it is a destructive technique, it's mass spec. You are injecting molecule into the detector and your sample is gone. So that's one. And another would be, so there is almost no way we can do live imaging. It's unlike the fluorescence imaging. You have to fix them and then put into the vacuum or outside, and then inject the mass spec. So that would be the major limitation, that there is no way mass spec imaging can overcome some other technology.
Yeah, definitely so. Then that's actually a really good point to bring, is that everything is actually not going to be in vivo at that point. You're exposing it to organic solvents, whether these are strong acids, bases, anything in between, or actually embedding them into different cellulose. It's really hard to go ahead and actually do that in vivo. So you have to design your experiments, whether they're time point studies or anything in between, that leaves large gaps where modeling or maybe some other techniques can actually go ahead and fill in a lot. So thanks for playing along with my little audible. I think it's very important that, especially for a lot of the people here.
Absolute quantification of technique, like, and I always have a hard time, like, quantitative part of mass spec imaging. I mean, that can be overcome probably, like, and it will, I mean, we're going there, but there are still like limitations there.
Oh yeah.
Especially because in heterogeneous tissues, like, intensity of signal depends on the matrix. So if you have like really heterogeneous sample signal that is higher in one part and lower in another, doesn't necessarily mean that here you have more than here. It just, like, that matrix here kind of suppress signal there. So yeah, that also kind of one of the
big disadvantages
I find. And maybe also, like, in terms of MALDI, it's also like biased technique.
Correct.
Towards some of the compounds. So
yeah, there's a really good example of this, of Kristen and Youngjin were talking about lipids, but if you have, say, phosphatidylcholines versus phosphatidylethynolamines, and if you have a stable concentration of the ethanolamines, but a differential concentration of the cholines based upon MALDI or nanodesi or anything, you can actually artificially inflate based upon the actual charge discrimination and the actual ionization efficiencies of each of these. So when we're talking about mass spec imaging, especially Fourier transform mass spectrometry, which we're indirectly detecting all of these samples, just because you have an M/Z 100 at an intensity of 1 and M/Z 200 an intensity of 1 doesn't actually mean that these are equivalent. So when we think about quantitative mass spectrometry imaging and other things within this, there's a lot of very unique aspects that we have to go ahead and take advantage of. And as Youngjin mentioned, a lot of this will be solved in the future, but right now it's one of those ambiguous portions of where we currently are. So in terms of relative quantitation within the actual spectra, that's something that, it's up for debate. I tend to lead on the side of we're relatively quantitative, but absolute quantitation is very, very difficult. So I don't know if anybody wants to add anything to that, or if we wanted to scoot on to the next question.
I meant to say there is the challenge that
Yeah.
we have that we would discuss in the next. But I don't think there is no good solution. If there is a local ion suppression, then that's a major problem right now.
Yeah, definitely so. Okay, so for our panel question number 3, I'm going to say, what is the biggest challenges? And I don't know, we did sort of like the disadvantages within this, but I want to really reinforce that there are a lot of disadvantages. There's a lot of advantages as well, but there's a lot of things that we can sort of start talking about. And I don't know if, you know, we might have just talked a little bit about this between this, but if quantitation is the biggest challenge you think that's facing MSI, or what would you say is like sort of our keystone for the next 10 years of like, what should we solve?
Yeah. I think quantitation is always a problem. For me personally, so my PhD is in biochemistry, so I care about pathways. And, you know, I think a lot of people have always said, okay, you plug it into KEGG, this is your pathway. I think we're now getting the amount of data we need to show that there's a lot that we don't know. And so using tools like David mentioned, where you're actually looking at co-localization of metabolites, specific proteins, specific enzymes, specific lipids, and starting to put them together into a biological context of when something, if you have a community that's degrading lignocellulose, you can see what specific enzymes are being secreted by the fungus at a target point, seeing what metabolites are coming off of it. Maybe the bacteria in that local environment are taking up those metabolites. They're then processing them to other things that are fed out to the system again. So those are the type of questions that I think we're getting to the point where now we can start addressing them and really getting to that biological knowledge that we need to inform systems, to perturb systems, to create systems for ourselves. So that's my passion, trying to figure out everything, how biology is doing it, understanding the complexities, and really pushing it to the next level.
Yeah, I think that's a very great point, actually, of the analytical methods are now at the point where there's not much that you can actually advance in terms of that, but it's now the biology that's a tricky point, which is where all of the fantastic EMSL scientists that are bringing this biology to us, we have all the tools and the tricks of the trade, but it's just answering the questions that we actually need, designing those experiments. So, John, I don't know if you have anything to add to this. I know we mentioned a lot about the disadvantages and the challenges before this, but.
Well, I guess I'll take Dušan's answer and cube it. You know, quantification and stuff, and it's possible in some ways with SIMS, but it's a lot of work and money.
Yeah, expensive experiments basically.
And then again, the sample preparation. So if you want to lock things in, we use resin.
Mm-hmm.
It's 99% carbon-12. You know, so you've lost your carbon-13 signature. We've thought about trying to buy carbon-13 labeled epoxies, you know, but that's going to be, you know,
expensive.
$10,000 a gram, or I don't know what it is, but it's going to be spendy. And then finally, you know, a problem that I run to a lot, you know, you showed this thing up here where we had all those bacteria, and you know, it takes like 1,000 bacteria to see this, just, you know, to kind of, or to just start to look at the population dynamics, and just getting segmentation around those individual cells is not a trivial problem. David said he found 40 algorithms and before he got tired in 10 minutes or something, but I'll bet you 39 of them don't work.
Yeah.
For my, you know, it's not a trivial problem.
Yeah. Just because something was published doesn't mean it's necessarily directly applicable to what your samples will actually be. It's a very big challenge. I know we talked a lot, but Young didn't do anything else to chime in on within this.
So I also agree that absolute quantification is definitely the biggest challenge, and probably the next one might be exact identification what these compounds are, because Christian mentioned because there is no chromatographic separation in mass spec, you have to do either in parallel metabolomics analysis, or you have to do LISA with LC. So currently there is no good way. Maybe ion mobility can help a little bit, but ion mobility, the separation dimension is limited, maybe up to 80 at the most currently. So And on top of that, if you want to do real structural analysis, you may want to do MSMS. So maybe parallel ion mobility MSMS may help. But at the moment, the biggest challenge is that I think it'll take another 5 years or more.
Yeah, because if we think about it in terms of metabolomics, there's probably 16 million metabolites within even just the plant kingdom. And a lot of those share the same mass per charge. They're the same molecular formula, they're stereoisomers or anything in between this. And when we're just looking at just the actual carbon, hydrogen, oxygen, sulfur, we really can't necessarily say that this is going to be R or S. So in terms of the actual biology that we can get out of this, we're somewhat limited in terms of the actual scope of what we can do. So when we give some of the actual users that have never seen this data prior, we can actually only say, well, we think this is actually going to be a sum formula lipid 34-1. We don't know the actual acyl chain widths or where these double bonds are actually localized to. So that's really one of the issues with a lot of this data in terms of the readability to do these advanced biologies. So I think with that, we'll scoot to our next one within this. And it's sort of a topical question of how might computational techniques, whether these be machine learning or artificial intelligence, might solve these problems? Can they solve these problems in your opinion?
Yeah, I think one of my main focuses is looking at, multiple images and putting them through advanced AI and machine learning approaches so that the algorithm can tell me what's either co-localized, anti-localized, and get that biological information that way. Right now what Kevin spends and Dušan can spend hours, there are tools to help, but I mean, there is a lot of manual processing and you can see so many pictures, and the human eye is great at recognizing patterns, but AI is better. So if you can feed all this information to it and say, this is the region I'm interested in, this little tiny micron region where there's something going on that I care a lot about. Who's there? What are they doing? And what are their partners? Who also is in high amounts, who's actually being depleted. So that's the type of information that we want to try to get to in the future by using advanced approaches. And it saves time and we can get through it a lot faster.
Yeah, no, that's definitely true. John, anything to add within this? I know you talked about segmentation. There's a lot of good things that possibly might be able to help you with that.
Yeah, I guess I'm going to pass on this question because it's not, I've done a lot of TOF sims in the past. And of course, everything that Kristen just said is applicable there, but and segmentation is a more difficult problem than you think it would be, but it's not really applicable to everything I do right now. So
yeah, oh, it's completely fair. Young Jin.
So there are a lot of people working on this area in computational people. I don't think there is no AI approach at the moment, but there are some machine learning approaches going on, especially also with clustering and like a cardinals and so. But what is missing would be so there are a couple. One of them would be in some. So there are many dimension reduction tools are available, like we have seen it from single cell transcript analysis and so on by analyzing to UMAP. Some people are trying something similar for mass spec imaging data. The problem is when they do that at the moment, the best way they do it is taking each of those single pixels as if they are individual sampled, and they just throwing into the reduction tool and then analyze their dimensions analysis. The problem is then you are missing all the spatial information, so where they are from. So that is absolutely missing right now.
Yeah.
So that's one. And also one issue is in any of this data analysis, statistical analysis, or machine learning, currently there is no good strategy for the normalization. Of course, normalization is a general problem in multi-mass spec imaging because depending on the sample types and your biological questions, how you normalize will affect your result. But at the moment, people are just throwing into the machine learning algorithms, and what happens is the program will pick out whatever most abundant changes, which may not represent much about specific biology. Maybe your the molecule of interest might be very small, tiny peaks, and then the fluctuation of those peaks in special dimensions. Maybe the machine learning programs or any clustering program may not pick out those changes. So that's one problem. So some people are trying different ways of normalization instead of throwing in raw signals. Some people try logarithmic scaled data and then do it. But then there is another problem that how you differentiate with noise and so on. So there are a lot of problems. So that's definitely something data scientists can jump in, find the solution.
Yeah.
And another would be, so in case of clustering is probably one of the most powerful way of analyzing mass spec imaging data. There are thousands of mass spec images, and clustering program will sort it out which images are similar to each other. The problem is right now there is no good judgment of how many clustering is the best, right? So depending on the biological systems, if you cluster into 5 groups, that would be the best. If you cluster into 3 groups, that would be the best. But currently, just people use random approaches and then just go with it. So there is the same thing for machine learning. There are many different machine learning algorithms.
We have to train each of them differently, right, and based on your samples.
Just go, okay, random forest works best for me. And there is no region or no rigorous judgment why there is better. Yeah. So if there is some rigorous algorithm or some a measure of which one is the best, and so if you try 10 different machine learning algorithms, and then there is some global measure of why this one is better than the others, that would be great, so that it could be more globally applicable.
Yeah, and as analytical scientists, we always like to see metrics. But yeah, there's a lot of potential within this. But yeah, it's something that we're going to have to develop over those next 10 years. It's really one of those big problems that hopefully over the next decade, a lot more data scientists will come into the field and be able to apply a lot of these really nice techniques that have been done for a lot of the data science, but now into the actual biology. Compressed sensing is also another one. FTMS can be very slow, up to 10 seconds per pixel. So there's also some aspects of trying to actually see, can we actually use ML AI in order to actually reduce the time spent for each of these? So lots of different perspectives, problems that we can actually have, and some of this can actually be applied directly to it. I think with that, we think we have a little bit of time left in order to address any questions that possibly the audience might actually have, whether that's online or whether that's within the room. Is there any questions for our distinguished panel within this?
I can make one more comment too with the AI. It just got me thinking. But you know, whenever I work with the data scientists here, like let's use AI. They're like, you don't have enough data. They're like, we can do machine learning, we can do random forest, but you don't have enough data. And the data you have, you know, maybe you have data that's been collected over 3 years. That's amazing. But did you put in the necessary things to make sure that it could be compared across 3 years?
Correct.
We didn't before. We're now aware of this. And I think EMSL can make a big impact in this, where we can generate huge amount of data that can be comparable across years. So we can get that noise level in there, and that we can do true AI, a lot of the visualization approaches that we can't do right now. So it is an exciting time to have the data scientists here that can inform us of what we need, so that, you know, in a few years we have everything to really make that big impact.
Well, that's actually a really good point too, because quality assurance, quality control, especially when you're talking about different sample preparations, different biological replicates, there can be a lot of variants that you can actually have. So that's a very, very good point in order to make a lot of strides.
I think I want to piggyback on that comment too. We also see a lot of effects with sample type itself. So even if you've got some benchmark study where you've determined the optimum, let's say you go an unsupervised approach, so you don't even really have data that you're feeding the model to train the model and test it up. Even with an unsupervised approach, you could see effects where it works really well with one data type and not with another. And that is something we see all over the place, even like building metabolomics libraries. When you're just trying to identify metabolites, there are specific sample type effects, which may stem from the different sample preparation methods, but we don't really know. And that is definitely a huge barrier I've seen. Yeah. That's why we always ask you for more data. That's all you need to do to be a data scientist, is just ask for more. Follow me for more tips. Okay, is there any questions from the audience?
John, you had a comment as well?
I have a comment, something to think about. So during my PhD work, I became aware that, and it's an opinion really, that everything's controlled at millimeter scale or smaller. And when you think about that, understanding these different problems at different scales becomes important. And I would argue that these systems are not only fractal, but they're likely chaotic. And so what that means is you've got this complex modeling system, and you come out with an answer like this at 3 decimal places, but if you go to 4 decimal places, you might get this answer. And so it becomes a really very difficult problem. And so, I really like that really small scale, because that interests me, but I think we need to understand this at multiple scales and multiple dimensions, chemical dimensions and physical dimensions as well, and temporal.
Yeah, temporals. That's one of the biggest challenges, where I think ML and AI especially has a large potential, because we're destructively analyzing these samples. We have a time point, maybe at 1 day or 1 drought period. If we watered our plants after biotic stress or abiotic stress, and it was at 50% water for several days, and then we actually have that rehydration step, we can't actually analyze at every single point. We always want more data within this, but we might only get 1 out of this. So that's actually something that's a large challenge that we might be able to start inferring in between the lines. So I don't know if this spurred any extra discussion or any thoughts within anybody. I know there's some people out here that say they're non-experts, and that's actually it's perfectly okay within that, because there's a lot of things. Everybody up here will actually say they might be called an expert, but there's always continual learning within this, and bridging into the first gaps of mass spectrometry imaging. There's a lot to be digested. So, but let's see. Okay. Well, with that being said, I think, yeah, I'm not too sure how to see if there are any online questions and monitor that. Yeah, we'll have to see. Oh, that's completely fair within that. So with that being said, we might be able to just conclude just a little bit early. It's just a couple of minutes within that, and we can give a great round of applause for our panelists who took time out of their day. So thank you guys kindly very much.